Hello, my name is Ruchin Patel, and my project is on evaluating unknown proteins. So, even though the human genome has been sequenced, it hasn't been fully annotated yet, and many proteins have yet to be identified, either in form or in function. So, for this project, I'll be taking 5 proteins identified in Daniels et al. As being in chromosome 16, p13.3, that are in section 1 of 8 as identified by the paper, and that are labeled as unknown or having an unknown function. So to characterize and identify these proteins, I'll be using 2 different methods. The first is homology and domain identification to identify function. And so for that, I'll be using NCBI BLAST, so both BLAST-P and T-BLAST-N. I'll also be using interpro scan and scan prosite. And I'll then be using structure prediction to identify form. So for here, I'll be using Swiss model to do homology-based modeling and Robetta to do ab initio modeling. And I will be comparing those models after they're created. So for the first protein, we can see the BLAST results for the BLAST-P up at the top. And so that is for a protein called HCG1742852. So that was the sequence that the BLAST returned, and that is for a WASIR1 gene. The T-BLAST-N returned WASIR1, WASIR2, and 3 different predicted tubulin beta 8s, but they only had 33% query cover, as you can see down here, the small blue bars, and they had lower scores. So they definitely are not as strong homology predictions as the WASIR genes. So the interpro results only gave disorder predictions, which is not very useful, and prosite did give some results, but they were all high probability sequence hits, and they were all phosphorylation or N-myristylation sites, so hard to really identify function from there. So looking at the modeling here, the Swiss model, there were 2 different templates that covered different parts of the sequence, but both gave poor GMQE and identity percentages. So those models are in the corner here. You can see the green and blue model here for the first one, and the orange and yellow for the second one. The Robetta model had a 0.43 confidence. You can see that here in the rainbow. And then if you look in the corner, the gold is the Robetta model. And you can see that neither of the Swiss models really have any sort of consensus with the Robetta model. And given the higher confidence of the Robetta model, it is much more likely that that one reflects better what the protein might look like. Moving on to the next protein. Here we see that BLAST-P gave conserved domain results. So here it gave a ubiquitin domain. And then looking at the protein sequences, all the protein sequences were U11, U12, small ribonucleo 25 kilodalton proteins. I just picked the top 10 for the graphics. And then doing a T-BLAST-N, again, they were all U11, U12, small ribonucleo 25 kilodalton proteins. The interpro family results, again, were the same, and the domain did correspond with the BLAST result of a ubiquitin domain. And the prosite also gave a ubiquitin 2 domain. And it also did give a protein prenyl transferase alpha subunit repeat identification, but it was low confidence. And given all of this consistency, it is very likely here that the protein in question is a U11, U12 small ribonucleo protein, 25 kilodalton protein. So now looking at the modeling, the Swiss model, it The main template was, as expected, the ribonucleo 25 kilodalton protein, and it has a decent GMQE score and high identity. The Robetta result has 0.79 confidence. You can see again Robetta in the rainbow on the left side, and the Swiss model on the right side. And looking at the comparison between the two, again Robetta in the gold, you can see that there's high similarity with nearly identical secondary structure. So both methods are giving us very similar results. Now moving on to the third protein. Here our BLAST-P gave us a conserved domain again. We have rhomboid serine proteases near the N terminal and the rhomboid domain near the C terminal. And the sequences that it gave for conservation were rhomboid 5 homolog 1s and inactive rhomboid protein 1s. And they all had very similar level E-values and identities and coverage. The T-BLAST-Ns, that one gave rhomboid 5 homolog 1 as all of the returns. So the interpro result, again, identified it as a rhomboid in the rhomboid serine protease family and identified a rhomboid domain. It also did identify transmembrane regions, so I went ahead and did a Phoebus analysis to double check. And as you can see, it does identify that the rhomboid domain is consisted of 6 transmembrane helixes, which is what the rhomboid domain is typically consisted of. And the pro site results, both these hits were low confidence. One was an arginine-rich region. The other was a bipartite nuclear localization signal. Again, low confidence, not really the best to base function predictions on. And now looking at the modeling, we can see that the Swiss model gave a rhomboid protease, but it did give even low scores, even a low Q mean at negative 4.96, where negative 4 is typically the cutoff. And Robetta had a 0.34 confidence for its structure that it created. Again, Robetta here at the bottom, and the Swiss model here on the right. And when you compare the two models, you can see that the region that the Swiss model covers is over here on the left. And the secondary structure is similar, but there are some significant differences. So there is some agreement, though there is some major disagreement, but that is mostly on the ends of the Swiss model. So, whereas the center of the model is much more consistent. Now, moving on to our next protein, we have only one BLAST-P hit, and that's isoform CRA under a D. That is a FAM234 A protein, but the T-BLAST-N does not return any of those. Instead, it returns an interleukin-9 receptor pseudogene, and as you can see here, only 33% cover, very low E-value. And then the interpro, all it returned was a transmembrane region, and again, I did a Phoebus just to double check, which it also did return that. And the pro site, again, only returned high probability sequence hits. And these were myristylation sites and phosphorylation sites. So there's not really much to base any function predictions here. Modeling, you can see two different models here on the right. And the Robetta did give a 0.54% confidence model. But when doing the comparison, the Robetta did have some similarity with the N-terminal protease model, which is the one on the top, but the type 1 topoisomerase model did not have any sort of similarity. And moving on to the last one, this one gave FAM234A hits on blast P and as well as T blast N. And the interpro, it did identify a homologous superfamily, quinoprotein alcohol dehydrogenase, but it also did identify FAM234A in there. Again, this one did identify transmembrane regions, so we did do a Phobius that did corroborate. And then looking at prosite, there was one low confidence hit of coagulation factors 5-8 type C domain. And then when doing the modeling, we saw one hit for endolase CSW. Again, not too high scores for GMQE or identity. And we did get the Robetta with 0.42 confidence. And when doing the comparison, there are some similarities, especially a few beta strands that look to be in the same in similar positions, but there are also very large differences. And so obviously there's a lot of predictions we can make about function here and looking at the form. And I'll be coming to some more solid and concrete conclusions in my paper. Here are the references with the main paper and the references for all the tools that I used.